All right, well, let's kick things off. First of all, thank you so much for joining and welcome to the inaugural Biosensor seminar. This is, of course, a relaunch. We attempted to launch this series last month and had some issues with the Zoom bomber, which was my first experience, and I believe the first experience of anyone on this panel with the Zoom bomber. I think we all believe that those days were, we left those behind a couple of years ago, but that turned out to be wrong. So as we begin, I want to mention that the seminar is being recorded and it will be posted on YouTube on the Biosensor Seminar Series YouTube's channel. You will be able to find a link to that YouTube channel on our webpage. So our goal with this inaugural meeting is to first of all describe the mission of the seminar series, to introduce the organizing committee, and to briefly discuss the scope of what we hope to include with this seminar series. Simon, if you could go to the next slide. So our mission with this seminar series is to bring together a community of scientists and bioengineers that are building genetically encoded sensors so that we can share our research findings. We really want to emphasize here early career research. So if you're a graduate student or if you're a postdoc, if you're a trainee, if you're just a new PI, those are the folks that we really want to spotlight. We want to use this venue to facilitate collaboration and discussion with other folks that are like-minded and interested in building genetically encoded sensors, as well as to teach fundamental and new concepts to researchers in this growing field. So perhaps you only have a passing interest in biosensors, but you want to learn more. We aim to be here for you as a resource so that you can learn the state of the art in the field of biosensing. So this seminar series was created for you, the community, and we welcome any feedback that you might have to improve this series. The format that we're going to be following for this series is a 30-minute seminar, the first Wednesday of each month at this time. And we also encourage everyone that wants to present or wants to, recommend someone to present. You can go to our website at biosensorseminar.org, and you can nominate yourself or you can nominate others. And so with that, I want to introduce the organizing committee. And so I'll start with my own introduction. So my name is Khalid Alam. If you go to the next slide, my background, I'm trained as a biochemist. I did my PhD at the University of Missouri, where I worked in the lab of Donald Burke, where it really focused on RNA structure and function. So I developed a software toolkit for next-gen sequencing analysis of aptamer selections, really focused, though, on again, functional RNAs and fluorescence activating RNAs. And so I was really interested in using RNA and building RNA based sensor systems. I followed that up with training at Northwestern University at the Center for Synthetic Biology with Julius Lux, where we developed an RNA sensing platform that we termed Rosalind, as well as doing additional work around cell-free biosensing broadly and developing genetic circuits, particularly transcriptional circuits, to really develop and fine-tune cell-free biosensor approaches. If you go to the next slide, all of that work, both as a graduate student, as a postdoctoral student, led me to the formation of StemLoop. So I'm founder and CEO of StemLoop. We're a company that's commercializing cell-free biosensor technology. Our mission is essentially to make it easy to measure small molecules. In addition to that, I'm also co-founder of a nonprofit called Chi-Town Bio, which has opened up Chicago's first public biotechnology laboratory. So if you're ever in the Windy City, come visit us at 6634 North Clark Street. And then finally, if we go to the last slide for myself, what I'm interested in broadly, there's a sort of a number of different topics, including the bioeconomy broadly. If we sort of narrow in from there, I'm really interested in synthetic biology, so being able to engineer and design these systems. I'm a big fan of using cell-free. I think biology is really complex. And if we take away the complexity of life itself, we end up with just a vat of goo that has metabolic capabilities. So I'm really keen on cell-free systems, and in particular, using allosteric transcription factors as biosensors in cell-free systems. Also quite keen on functional nucleic acids and using all of these things in sum to create point-of-use sensor and diagnostic applications. As for my why, I fundamentally believe a few things. One is that biotechnology has transformative potential for both the people, for people and the planet. I believe that our time to act is now because our ability to read, write, and compute with DNA is completely unprecedented compared to any other time in history, and that biology is the best analytical chemist. And I'm hoping to learn more and share more on that topic moving forward. And with that, I will pass it off to our other organizer, Eric.
Hi everyone. So my name is Eric Hanko, and my main background is in metabolic engineering. So for more than 10 years now, I've been working on engineering model and non-model organisms to produce platform and fine chemicals. Um, so I did my PhD at the University of Nottingham, and the main focus of my work was to establish Cuprovitus necator as a biocatalyst to convert waste gases into platform chemicals. However, we soon realized that Cuprides was able to metabolize some of the compounds that we're trying to produce. This is basically how I got interested in biosensors, just by trying to figure out the mechanisms that are involved in sensing and responding to these compounds. So as a result of that, I developed and characterized a range of different biosensors for platform chemicals like itaconic acid, 3-hydroxypropionic acid, and some aromatic compounds. Next slide, please. So after my PhD, I then moved to Manchester. So currently I'm a research fellow in biomanufacturing at the University of Manchester. I'm still interested in mining biosensors for user-defined molecules from bacterial genomes. But I'm also working on using biosensors to evolve pathways to produce unnatural compounds. Next slide, please. So now that we have all these cool sensors, uh, one thing that I'm really interested in is what can we do with them. So I'm really interested in novel applications of biosensors and how we can implement these biosensors in genetic circuits to dynamically control metabolic pathways. For the reason why I'm here, first of all, I want to hear more about all the fantastic research that's going on in the field. But I'm also excited to make just new connections and to really help build a biosensor community. Next slide, please. So Megan unfortunately can't be here today because she's at a conference. So I'm going to introduce Megan. Okay, so Megan, she did her PhD at Berkeley, where she became interested in transcription factor evolution. There, she studied bacterial two-component systems with transcriptional activity and specifically explored possible evolutionary pathways that allow them to expand in sequence space. Next slide, please. So now Megan is a postdoctoral researcher at UCSF, studying transcription circuit evolution in fungi. She's interested in exploring new pathways that allow regulatory networks to evolve regulation of new sets of genes. Next slide. And then Megan's interest in biosensors stems from a deep fascination in transcription regulation. She hopes to see the seminar cover basic biochemistry, evolution, and she wants to note that she would love to see those projects discover or engineer heteromeric protein-protein interactions, which are commonly found in transcription circuits across the tree of life. And with that, I'm going to pass over to Simon.
All right. Thanks, Eric. So yeah, my name is Simon Dolsnitz here, and um, I joined the PhD labs of Andy Ellington and Hal Alper at UT Austin. That was back in 2016. And I think my main motivation at that time was I really wanted to engineer bacteria and different organisms to produce chemicals, really that biomanufacturing angle. But in the lab of Hal Alper, because that was his lab's main focus, I realized that one of the big bottlenecks of engineering systems to produce molecules is the analytical step of testing whether your organism produces enough or the right type of molecule. So from that, I kind of did this joint PhD with Andy Ellington and Hal Alper, where I really settled on using transcription factor biosensors for high throughput engineering of strains and enzymes and pathways. And so my main focus is on using directed evolution to customize the specificity of transcription factors for different interesting molecules. The ones I initially focused on were therapeutic alkaloids from plants, as well as terpenes. And in particular, I like to use these, what we call these very malleable or fungible biosensors that can be evolved in different directions for specificity to different molecules. And then also during my PhD, I use those sensors and high throughput screens to engineer improved enzymes. So right now, I am a postdoctoral fellow at Harvard Medical School in the synthetic biology hive. This is kind of co-run by Mike Springer and Pam Silver. And the main focus of my work currently is just generally to produce a more systematic way of engineering custom biosensors. So a lot of the way where a lot of the strategy to make biosensors is kind of ad hoc. You know, you just happen to find a sensor and you cobble together some library and engineer it. So a big focus is on using massively parallel reporter assays to collect large data sets of a bunch of variants and how they respond to different molecules. And then using that data to do predictive engineering of targeted specificity and sensitivity. In terms of the application, I'm mostly focused on engineering sensors for pharmaceutical manufacturing, in particular for enantioselective reactions. Why I'm interested, I'd say at the end of my PhD, I kind of felt like this character on the right, where I engineered a thing and I said, "Hey world, this is a thing I made." And you kind of felt a little isolated. And I think what I realized was missing was this sense of community where you could share your research, where you could learn from other people and really get good feedback and just grow together as a community. So main motivation is just to bring that community together and build cool things. The topics I'm interested in are prokaryotic transcription factors, in particular, engineering their ligand specificity and applying them to enzyme engineering. So I think that wraps up the introduction of the committee members. Now I just want to shift gears a little bit and talk about scoping and cover what topics we're hoping to kind of cover in this seminar series. It's still early on. We're still very adaptable, but at least as an initial starting point, we're kind of thinking about two discrete areas. So first, our general focus is on genetically encoded small molecule sensors. This could be proteins, this could be RNA. We're really just interested in how nature and biological systems measure small molecules. So breaking that apart into two different focus areas, there's a more fundamental avenue or questions we'd like to explore, which is how do biological systems sense chemistry? And then the more application focused angle, which is how can we engineer sensors for practical applications? And so now I'm just going to go into a really brief history of some of the big milestones that have been made in the past few decades, leading up to where we are now and what we would like to talk about in this seminar series. So you can track back the history of how biological systems sense chemistry to the famous 1960s work by Jacques Monod on the operon model, where the lac repressor was identified as the kind of component responsible for this dioxic shift of E. coli being able to metabolize lactose. Following that, there's some seminal work in the field of like structural biology with the advent of X-ray crystallography. So the structure of the phage repressor was elucidated in 1967, as well as, or actually that was evidence of repressor binding to DNA. And then in the 80s, several structures came out of regulators that are bound to DNA, which provides this mechanistic basis for gene regulation. Following that, some big work from the Breaker Lab found that not only proteins, but also RNA is used for small molecule sensing in organisms. And more recent work with the advent of cryo-electron microscopy has been able to elucidate large complex structures of proteins, RNA polymerases, and DNA together to find out how exactly these components work together to modulate transcription and translation upon binding to a small molecule. So leads up to right now, which is what are we interested to talk about? One side is mechanism. How do biological systems bind to and respond to small molecules? What kind of chemical motifs are used, as well as characterization. I think there's some statistic around that's like about 10% of an organism's genome is dedicated to regulation, and a huge vast majority of that is still to be characterized. So there's a lot of work to be done on that side. Now shifting a bit to the engineering side and application. A a big application of genetic sensors, genetically encoded sensors, has been the use of inducible systems. So using classically TET-R or LAC-I to regulate the expression of genes. One of the big seminal papers was from 1987, where the LAC-I repressor was used to regulate expression in mammalian cells. Since then, in the decade following, people have used it for expression in non-model bacteria, as well as plants and insects, and even monkeys, living monkeys. In 2000, a lot of people attribute that year to the year of synthetic biology, where these regulators were used together to form more complex gene circuits. And with that comes several different interesting kind of logic functions that can be done. One case was an oscillator circuit, and then the other was a toggle switch, but many more have come out since then. 2006 was a big year in terms of application. On one side, the first group used a transcription factor to create a high throughput screen for screening for improved enzymes. And the work from Francis Arnold's group actually used directed evolution to alter the specificity of transcription factors, the ligand specificity. So that opened up now a huge area of potential research to explore high throughput enzyme engineering and also custom sensors dedicated for certain screens. And more recent work from several different groups, including Khaled, is focused on the application and deploying of these sensor systems to create multiplexed inducible systems, to create diagnostics for environmental pollutants, as well as for engineering enzymes that actually go into clinical trials. So we're at a very exciting time in the development history of this work. Things we're interested to talk about on the application side, one would be engineering of novel sensors with altered specificity or interesting properties. The second thing would be applications, so incorporation of sensors into things like diagnostics or high throughput screens or any other application you can think of. We're also interested in exploring different methods for the high throughput or otherwise in-depth characterization or discovery of genetic sensors. So with that, I just want to wrap up and say thanks for joining. We're really excited to kick off this seminar series and hear from you about what research is going on and your feedback and thoughts. Our next meeting will be in 1 month on April 2nd at 12 p.m. Eastern Time. And please feel free to sign up for presenting in this seminar series. We're excited to hear about your work and what you have to say. Any wrap-up thoughts, Khaled and Eric?
I'll just say that super excited to start building this community. As Simon said, in my own personal experience, right? We develop these things and then we get them out into the world, and it does very much feel like we're alone. And then with time, you sort of start to see your papers being cited. You start to see, sessions at conferences that are focused on this topic, and then you realize you're not alone. So, but really looking forward to building out this community. We certainly all have topics that we're interested in. We're trying to keep the scope of the Biosensor seminar series relatively flexible. So if there are certain topics, if there are certain presenters, or if you yourself want to present, please do let us know. I'll just remind everyone, bookmark biosensorseminar.org, follow us on LinkedIn, and look forward to the next meeting in a month.
And I would just like to repeat what Simon said, so I can already see in the chat that we've got a lot of people with a lot of interesting projects. So please sign up if you want to present, or if you can nominate someone else to present to hear about their work. So please take advantage of that. And then I hope I see you next month.